defining chromosome regions. If we take the example of a typical chromosome consists of centromere that is present in the middle, centromere, and it consists of two arms. This is the arm of the chromosome and this is the second arm of the chromosome. So a typical chromosome consists of a centromere and two arms. One of the arm that is usually short in nature, that is called P arm, while the longer one is called as Q arm. So there are two arms of each chromosome. Each of these arms, they have the regions. The larger fragments which are present on the P arm, they will be written as region 1 and region 2. We can say that this region can be written as 1 and this region of the chromosome can be written as 2, region 1 and region 2. Same is on the lower side. In case of Q arm, we can see that region 1 and region 2, they represent to this part of the Q arm and to this part of the Q arm. So on P arm, there are two regions, 1 and 2. Same is on the Q arm, region 1 and region 2. Now each of the region that consists of bands, or within the regions there are bands. Within a region 1, there is only one band. There is region 2. Within the region 2, there are two bands, band 1 and band 2. So region 1 consists of a single band, while region 2 consists of two bands. Same is here in case of Q arm. We can see that region 1 consists of two bands, band 1 and band 2, while the region 2 consists of band 3 and band 4 are within the region 2. Within the bands, there are sub-bands. If we see here, within band 1, we can see the sub-band is 1, 2, 3, 4 and 5. So these are called as sub-bands. We can conclude that sub-bands are present within the bands and bands are present within the region. Region are the larger fragments we can see with the help of the microscopes during performing the karyotyping. If a gene is located, for an example, on chromosome 17q, A11.2, then how we will identify that which one is the band or which one is the region where that gene is located? Look at the example. Let's say this is chromosome 17 and gene is located on 17q11.2. So we will say 17 is for the chromosome, Q is for the arm, that is Q arm, and 1.2, 1 is for the region, and this one is for the band, and this 2 is for the sub-band. So in this way, we can identify that where a gene is located. We can see that bands are present within the bands. Just to understand that how bands are present within the bands, if this is a band, here we cannot see further fragments which are present in this band. But when we will magnify the microscope, then we will see that there are further bands within this band. So this band consists of 3 sub-bands, 1, 1, 2 and 1, 3. But again, when we will magnify this band, we will come to know that again there are 2 sub-bands, 1.1 and 1.2. This band is 1.2 and this band is for 1.1. Chromosome identification can be done based on 3 different things. Chromosomes can be identified based on their size. Chromosomes can be identified based on the position of the centromeres, and chromosomes can be identified based on the bending pattern which is present on the chromosomes. So this is the complete chromatogram of the humans. In case of this chromatogram, we can see that how bending patterns are represented while describing the human chromosomes. We will just take the example of one of the chromosome, that is chromosome 1. In this case, this is P arm, this is Q arm. Each of the arm has regions. Region 1, this is region 1, region 2, region 3, region 4, and each of the region has bands. Then these bands, they further have sub-bands. So in this way, chromosomes, they have been divided into regions, bands, and sub-bands. Chromosomes have different regions, and these regions, they are labeled in the form of regions, bands, sub-bands, and with the help of these nomenclature, we can check or we can identify where our gene is located, or we can also write the position of a gene with respect to the bending pattern.